Amyloidosis, as simple, it is an accumulation of misfolded proteins around the cell wall, which causes damage to the organs whichever involved. What are proteins? Proteins are classified into two types. One is the structural proteins and the non-structural proteins. In amyloidosis, it is the non-structural proteins which is getting affected. So what are the three main types of proteins over here are? One is the immunoglobulins, second is the transthyretin, and third is the amyloid A. In immunoglobulins, it is the kappa and the lambda, that is a light chain which is involved in the amyloidosis, and transthyretin, which is a transfer protein which carries thyroid hormones and vitamin D to multiple organs. And amyloid A is an acute phase reactant protein, which means that whenever there is an inflammation, there will be a release of cytokines, and this cytokines will stimulate the liver to release the amyloid A, so that it can go and act on the specific organs for inflammation control. So these are the three types of proteins. So when there is a misfolding of all these proteins, it will just accumulate around the cells of the organs, and it will damage the organs. So what are the end organs? It will be the heart, liver, kidney, and even the bone marrows. So the symptoms will lie accordingly whatever organs are involved. So how to find this? This detection can be done by detection of protein. So it is a protein misfolding, so obviously we have to detect the protein. So what is the only test we can do as a screening test? That is the serum protein electrophoresis, in which we can find that there is a high level of protein. So the next step will be the what type of protein it is. So there are three types. One is the immunoglobulin, kappa, and the lambda chain. So we have to check for the serum free light chain protein content. That is one of the tests, and also with the mass spectrometry we can find it. So this is one of the method. Transthyretin and amyloid A, we can't directly find it out. So what we can do is either by genetical markers we can find this transthyretin and amyloid A, or by biopsy we can use using the strains called as Congo red or the apple green bifringes staining, and find the excess amount of protein surrounding the cell, and we can confirm it that it is an amyloidosis. So this is how we can find it is an amyloidosis. So as a whole, amyloidosis is of three types: AL, ATTR, and AA. So AL is amyloidosis light chain, ATTR is amyloidosis transthyretin protein, AA is amyloidosis amyloid A. So these are the three types of amyloidosis. So these are the most common, and this is how we find the lab diagnosis of amyloidosis.